All right, so part 2 of hair analysis and forensics. So we're going to do a little recap. Remember, it can be considered class, where you could say it's a group of people, or individual. You can narrow it down to one person, but it can only be that way if you have the follicle, because in the follicle, it has the DNA, which can link to a person. Other parts are composed of the cuticle, cortex, and medulla. Macroscopic properties. Anytime I say macroscopic, it means I can see it with my eyes. I have long, brown, straight hair. That's macroscopic. Whereas microscopic, you can only see in under the microscope. That's the cortex, the medulla, and even the cuticle. All right. So there's 4 types of medullas. You've got absent, continuous, interrupted, and fragmental. All right, so what we're going to look at today is some more properties of hair and how they're used in the forensics and forensics in general. So human hair can vary from region to region on the body. So some places you could think your head, your eyebrows, if you are a guy and you have a beard, pubic areas, body. What I mean by body is like I've got arm hairs, leg hairs, and then you've got underarms. All those regions have different types of hair. Hair can vary from one source to another. So not only on the body, but one source to another. For example, different ethnic groups have unique characteristics in the properties of their hair. So you can see right here, look at this. This is when it's cut down. You can see this is elliptic and twisted. This is just elliptic, and this one's round. Right? So they're all different. And you can see some more down here with the amount on a person's head, the general strand shape. So hair examiners have identified some key physical characteristics that are associated with the hair of different ancestral groups, like European, Asian, African. On top of that, we are not the only things that have hair. Animals have hair. So there's differences between human and animals. That includes pigmentation, what type of color it is, the medullary index, which we'll discuss in a little bit, and cuticle type. So looking at this, look at the medullas on each of these. Like you can't even see the deers. It's almost black. And then this has like curly cues. This has like little spherish things. And the mouse just looks weird. And this is a human with a fragmental. So on top of that, some comparisons. Human hair has color and pigmentation is constant, so it doesn't change throughout the hair shaft. Via dyeing, but that's only if you dye, it's only the outside, not the inside. Whereas animal hair can have multiple colors on one strand. The medulla, it's a third of the shaft diameter for humans, and amorphous. That means it doesn't always, it's not, doesn't always look the same on each side. Animal hair is this like a wide variety of medullas, well defined within certain patterns. Like you saw the cat had those like little circle things. Those are well defined patterns. The rabbit was the curly cues going up. The mouse had like little liney things going up. And scales, that's on the cuticle. It's overlapping for ours. If you've seen any commercials with shampoos, they typically show you the cuticles. Ours overlaps each other, where animals, it's like a wide variety. Maybe imbricate, coronal, spinous, kind of looks like spines, so on and so forth. Like lots and lots of different types of scale structure. So medullary index, we did mention that. I just did a little bit ago. What the heck is that? It is the ratio of the diameter of the medulla to the diameter of the entire hair, known as the medullary index. So if that medullary index is 0.5 or greater, there is most likely animal. If it is 0.33 or less, it's most likely human. Here is the equation for the ratio. The width of the medulla, that's inside the hair shaft, and the diameter of the whole thing. So suppose a hair has a diameter, that means from here to here, of 110 microns, and has a medullary that measures right from here to here, only 58 microns. What is the medullary index of the hair? So this is the first thing, and then we got to figure out, is it human or animal? So the width of the medulla, which is right here, 58, over the diameter. So I'm going to plug the numbers in. And then I'm going to use my calculator, and we're going to calculate it out. You should get 0.53. And if you remember, human or animal, human is 0.33 or less. So this is probably an animal. All right, so going on a little bit further to the structure of hair. Hair is incredibly strong thanks to a protein called keratin. And keratin is a chain of amino acids that form a spiral helix. And keratin helixes are connected by strong bonds between the amino acids. So it's like all connected together. These bonds also make hair resistant to decomp. This is the huge thing. It's resistant to decomposition, which is extremely useful to forensic investigators because you can use it well after this person is dead and gone, whether by natural causes or not. So lab analysts use a variety of methods to analyze hair, and you did read about one the other day. Hair is collected by plucking, that's what this is right here, shaking, like shaking stuff out, and scraping. If there's, I'm assuming if there's adhesives or something, they've got to scrape it off. Adhesives are also used to collect hairs from victims or objects. Adhesives, very important word, adhesives. So that's if you were to take like a lint roller and going like this, get the hairs off using adhesives. Hair can then be analyzed using microscopy or microscopes or even chemical testing. So when microscopy investigations are used, a comparison microscope is used to make the comparisons between the medullary index of a suspect and the evidence hair. So it's the side by side. You can see the strands, you can see the medullas, and you can see if they match up. Where electron microscopes, a different one, not a comparison, this is an electron microscope, it provides more details, which you can like zoom in 50,000 times the regular microscope. So this could be like a regular microscope. When you zoom in even more, you could see those itsy bitsy scales. Like, it's crazy. So anything ingested or absorbed through your skin becomes part of your hair, which is why it's great to be able to analyze the hair. This is proven to be beneficial for forensic investigators because remember, it doesn't decomp very well. So tests can establish a timeline of the suspect or victim's exposures to toxins or even drugs. For instance, we saw or read about this.
For the British government exiled Napoleon Bonaparte, the defeated emperor of France in 1815. Now he would spend his remaining years on an island of only 47 square miles.
Sorry.
He would die on St. Helena at age 51. The official statement was that Napoleon died of stomach cancer. For some, the story of what happened on this island is a story filled with clues, pointing to conspiracy and murder. That the great Napoleon would die such an ordinary death at a relatively young age.
Sorry about the picture, guys.
If he died of cancer, was he so overweight? There is more to this story, some insist. Almost 150 years later, in 1964, the hair that Napoleon gave to Betsy Balcom was one of several lots tested at Harwell Nuclear Laboratory outside London. There, in a brand new experimental process, the hair was placed into a nuclear reactor and made radioactive, enabling researchers to determine if the hair contained any poison. The results: Betsy Balcom's memento of hair was found to have high levels of arsenic, 32 times more than what was considered normal. Measurements were taken of individual sections of hair. The results showed that the levels of arsenic rose and fell because as the hair grows, the people stores that the blood went down to 1. Along with blood, no one has been able to say conclusively how the arsenic got there. Many theories have been proposed. One suggestion was that there was arsenic in the green dye in the wallpaper in the bedroom at Longwood. Others have considered the idea that Napoleon used hair tonic containing arsenic, or that he took medicine that contained the poison.
Which hair analysis cannot demonstrate poisoning. Poisoning means intentional. It means there is a poison. What I can say accurately is that the hair corresponds to somebody that has been exposed to arsenic, and that the concentrations that we measured are very high.
Napoleon's doctors found a mass in his stomach the year before he died. At the end, Napoleon was losing weight. He also had lesions that were very familiar. What did the governor with?
I think that the poisoning theory is finished. It's in its last throes. Everybody has understood that it's not serious. So, it's time for Napoleonic historians to go back to the real problems. Political studies, social studies, economic studies, the legend of Napoleon, which was created at St. Helena.
Why has the debate about Napoleon's death persisted?
The facts are too clear. Too clear. Too many factors shows that he did die from poisoning. It would be stupid to believe in anything else.
In the end, on St. Helena, Napoleon became a man much larger than life, and therefore, a man whose death would never be easy to explain. The island of St. Helena. It's where the British...
So, after that, a little bit more about Napoleon. What they used was called neutron activation analysis. Remember that, NAA? This can be used to detect toxins and chemicals within the hair. This test can provide links between hair evidence and the circumstances surrounding the hair. NAA was the nuclear test that was performed on Napoleon Bonaparte. All right, so you guys read about Napoleon Bonaparte. The next thing you're going to do is complete the review worksheet. And if you have any questions, please don't hesitate to ask either via email or class tag. Besides that, I hope you guys have a great day.